Hello students, this is Dr. N. Bhushanam, Associate Professor from Department of Zoology, Maharani Science College for Women, Bengaluru. Hope you are all doing well during this pandemic time. Take care of your health, students. So in today's session, we shall learn another topic of genetics, that is cytoplasmic inheritance, a concept important for essay question. The objective of today's session is to learn the cytoplasmic inheritance in two different organisms, that is Paramecium aurelia and water snail Lemnia peregrina. The content includes cytoplasmic inheritance of Kappa particles in Paramecium aurelia and maternal effect in the coiling of shells, that is chirality, in the water snail, namely Lemnia peregrina. Well students, when we look into the topic of our discussion today, it relates to the concept of inheritance. The process of inheritance generally refers to the transfer of characters or traits from the parents to their children or offsprings. So this inheritance takes place through the genes or factors as described by Mendel, and this is majorly related to the genes of the nucleus, that is the chromosomal structures. So chromosomal genes are involved in the transfer of character from parents to the offsprings according to Mendel, and hence it is called nuclear inheritance. So these genes are present in the form of functional DNA in every cell. When we look at the DNA distribution in a cell, most of it is concentrated in the cell organelle of the nucleus in the form of the linear chromosomes. But some part of the DNA is also distributed in the cytoplasm of other organelles such as the chloroplast, mitochondria, etc. So DNA of the nuclear chromosomes that inherits from the parents to the offspring controlling the characters is called nuclear inheritance. So down in the picture when we look at is an example given by the Mendel where the tall character and dwarf character of their pure lines, that is homozygous parents, when are crossed we happen to get the tall variety heterozygous offsprings. So the character of tallness is passed on from parents to the offsprings through the genetic material present inside the nucleus in the form of chromosomes. So this pattern of inheritance we call it as nuclear inheritance. So as we have discussed, some part of DNA is distributed in the cell organelles other than the nucleus, such as the chloroplast, mitochondria, and this DNA is also responsible for controlling certain of the traits or characters which are important for the organism. Such type of inheritance pattern is called extra chromosomal inheritance or extra-nuclear inheritance. In simple words to say, the genes that are related to the mitochondria and chloroplast are also responsible for controlling certain of the characters which are very important for the animal. So, such characters get inherited from the parents to their offsprings. So, this is called extra chromosomal inheritance or extra-nuclear inheritance. The same is depicted in the picture down here. So, when we look at here in the picture, we find towards the left side a cell showing the female parent and towards the right side the male parent. When you look at the characters that got inflowed into their child, the zygote or the child, there father side organelle are other than the nucleus are not transferred. It is only from the mother cytoplasmic organelle characters gets transferred. So, hence it is called extra chromosomal inheritance. Students, in the extra chromosomal inheritance, cytoplasmic organelles since are involved in the inheritance pattern, hence the inheritance pattern is also called as cytoplasmic inheritance. It is also referred as non-chromosomal inheritance. It was called Correns in 1909 studied this type of inheritance for the first time in the 4 o'clock plant, technically called Mirabilis jalopa. So, down in the picture when you look at of the slide, towards the left side is the photo of Correns and towards the right side is the 4 o'clock plant called Mirabilis jalopa. So, in all sexually reproducing organisms, there involves a male and a female parent for the production of the zygote. It is through the production of the gametes respectively. So, these organisms will produce their gametes, male parent will produce sperms as gametes, its head contributes to very negligible amount of the cytoplasm. So, a head of the sperm will have very less amount of cytoplasm contributed by the father side. But the females, the mother produces the egg or the ova as gametes, and these gametes will contribute to the high quantity of or high amount of DNA along with their cell organelles. So, the DNA present in the cell organelles which are responsible for controlling certain of characters in the organisms are called cytoplasmic genes, or in simple terms they are called plasma genes. So, total number of plasma genes that are present in a cell of an organism is called as plasmon. So, this is the major reason why the progenies or the children will generally resemble their mother very much, because mother will contribute to the most of the cytoplasm for the zygote. So, mother's characters are inflown into the child. So, this is what we call it as cytoplasmic inheritance otherwise. So, the same picture will explain the concept of cytoplasmic inheritance here too. So, this process of influencing plasma genes, a particular trait of their child or the children, is generally referred as extra chromosomal inheritance. In other words, the mother's cytoplasmic genes will influence the characters of the child directly. So, character of the mother will be passed into the child through the cytoplasmic genes. This is called as extra chromosomal inheritance. So, there are many synonyms, that is terms with same meaning are used for this concept of extranuclear inheritance. As nuclear chromosomes are not involved in the transfer of traits from the parents to the offspring, we call it as extranuclear inheritance. It also does not follow the Mendelian principle, hence we call it as non-Mendelian inheritance. It also refers to the DNA of other cell organelles, hence it is called non-chromosomal inheritance. So, cytoplasm of mother will contribute much in the cytoplasmic inheritance, hence it is called uniparental or maternal inheritance. We also call it as extra chromosomal or cytoplasmic inheritance. So, these are all different names that we give for the concept of cytoplasmic inheritance. The important features of cytoplasmic DNA or cytoplasmic genes are as follows. Otherwise, it is a comparison that we bring about the DNA present in the cytoplasmic region as well as nuclear region. So, this is the comparison between the plasma genes and nuclear genes overall. So, point number 1 is related to the cytoplasmic DNA. As we know, cytoplasmic DNA is present in the cell organelles such as chloroplast as well as mitochondria. So, cytoplasmic DNA is majorly distributed in the organelles of chloroplast and mitochondria. The second important point is the shape of cytoplasmic DNA is usually circular and double stranded compared to the linear molecule of chromosomal DNA related to the nucleus. Point 3 is synthesis of cytoplasmic organellar DNA, that is of mitochondrial DNA, is chloroplast DNA, is continuous throughout the cell cycle. So, as the cell divides and redivides, the DNA content of the mitochondria as well as chloroplast also divides. But this is not the case with the nuclear DNA. Nuclear DNA will divide or will be synthesized only during the stage of S phase of the cell cycle, that is synthetic phase. Point 4 is both cytoplasmic and nuclear DNA shows a common replication process called semi-conservation process or semi-conservative replication. Down in the picture, when you look at the blue color line indicates the parent or original template of DNA and red color indicates newly synthesized daughter templates. So, both are retaining one strand of the parent and new strand is synthesized. This is what we call the semi-conservative replication. Point 5 is both the cytoplasmic and nuclear DNAs undergoes the process of mutation. As we know, mutation is a sudden change in the DNA sequence or nuclear sequence of a gene. So, this sudden change can happen both for the plasma genes as well as nuclear genes, as what you see in the diagram down here. Point 6 is relates to cytoplasmic genes or plasma genes of either mitochondria or chloroplast codes for the proteins. So, DNA undergoes transcription to synthesize mRNA, mRNA will undergo translation process to synthesize the protein. So, the process of central dogma principle will hold good for the DNA of the cytoplasmic genes also, like that of the nuclear genes. So, this is the picture that depicts it. Point 7 is both the types of DNA, that is cytoplasmic DNA and nuclear DNA, can be synthesized in the laboratory conditions. So, in vitro synthesis of cytoplasmic DNA and nuclear DNA is possible. Okay. So, by using their complements. Well, students, after we understanding the characteristic features of cytoplasmic DNA and comparison of it with the nuclear DNA, now we shall draw the major differences between the DNA of cytoplasmic region and nuclear region, in simple cytoplasmic genes and nuclear genes. The first difference is related to their location. We know cytoplasmic DNA is located in the cell organelles of mitochondria and chloroplast, whereas nuclear DNA is present in the form of chromosomes inside the nucleus. Second difference is their shape or form. The cytoplasmic DNA is generally double stranded circular DNA, whereas the nuclear DNA are generally said to be elongated or linear DNA. The third important point is related to their synthesis stage. So, as we have seen, cytoplasmic DNA is synthesized throughout the cell cycle stage, whereas nuclear DNA is synthesized only during the S phase of the cell cycle. The fourth important difference is that the replication site. We know replication of the DNA occurs both in the organelles of chloroplast and mitochondria corresponding to the cytoplasmic DNA, but the nuclear DNA will replicate in the chromosomal structures. So, these are the major differences between cytoplasmic DNA and nuclear DNA. Well, students, after we understanding the concept of cytoplasmic inheritance, different synonyms that we give for it, and characteristic features of the cytoplasmic inheritance related to cytoplasmic DNA. Now we shall understand two important examples that are specified in your syllabus. The first one is cytoplasmic inheritance pattern of Kappa particles in paramecium. So, it is one of the most striking and spectacular example that we find in paramecium aurelia, which is a ciliated protozoan. Down in the picture you can see how paramecium of the strain aurelia is seen. There are many strains of paramecium, but paramecium aurelia will carry a symbiotic bacterium called as Sedibacter or Pseudobacter. So, when symbiosis happens between bacteria and paramecium, you find those bacteria inside the cytoplasm of the paramecium appears as particulate structures. So, these particulate structures are called as Kappa particles because they stay darker. So, these Kappa particles are nothing but the symbiotic bacteria called Sedibacter with that of the paramecium aurelia. So, these Kappa particles will divide, multiply inside the paramecium cell. It was in 1938, T.M. Sonneborn has reported two strains of paramecium aurelia, one with the Kappa particles, another without the Kappa particles. So, Paramecium aurelia, whose cytoplasm contains Kappa particles, are called as killer strain or killers. The one which do not have these Kappa particles are called sensitive strain. Down in the picture, when you look at and of the slide, towards the left side is the foot of the sonoball, and towards the right side is the paramecium with the Kappa particle indicating killer strain or killer. So, what are these Kappa particles? As we know, they are the symbiotic bacteria of Cydibacter, which is of 0.2 to 0.4 micrometer long, dark stained particles, which are with a range of 400 to 600 number per organism. So, if you could see in the picture down, those particles that we find down are called as the Kappa particles inside the cytoplasm along with the nuclear content, nuclear and cytoplasmic content. So, we know Kappa particles are found in the killer strains of Paramecium, and these Kappa particles produce a toxin. The bacteria will produce a toxin called paramecin. This paramecin comes out of the cell of killer strain, enters into the strain that do not have Kappa particles in them, called sensitive strain. So, once this Kappa particles enters into sensitive strain, there it kills the sensitive strain, damages their physiological activities. So, what is the importance? Sensitive strain will be sensitive for the paramecin secreted by the Kappa particles, whereas killer strains are capable of withstanding the part of the effect of paramecin. At the same time, paramecin secretion or production is seen in the killer strain with the help of Kappa particles or the Cydibacter, symbiotic bacteria. So, the toxin gene for the synthesis of paramecin is present on the bacterial plasmid DNA, which is nothing but an extra chromosomal DNA. So, every particle of bacteria will have an extra chromosomal DNA called bacterial plasmid that will in turn have the toxin gene for the synthesis of the paramecin. So, production of paramecin is dependent on the Kappa particles. Now, so production is related to Kappa particles, that is the Cydibacter of the paramecin. So, paramecin production is related to the Kappa particles or Cydibacter bacteria, symbiotic bacteria. Whereas, the multiplication of Kappa particles in a cell of paramecium is controlled by a dominant nuclear gene or allele, namely capital K. So, killer strains will have capital K allele in its dominant form, which is responsible for the multiplication of Kappa particles that are already there in the cytoplasm of the killer strain. So, it could be either homozygous K or heterozygous K. If capital K allele is there, they are responsible for the multiplication of Kappa particles if the Kappa particles are there in it, in the cytoplasm of the paramecium. So, what is the speciality of killer strain? Killer strain should have two major features. Feature number 1 is they should have Kappa particles in them. Feature number 2 is they should also have capital K dominant allele in them, either in the homozygous condition or heterozygous condition. When these two characters are fulfilled by a paramecium, such paramecia are called as killer strain. If any one of this is absent, they become sensitive strain, and the sensitive strain will have the effect of paramecin on them. So, absence of capital K or dominant allele will not make the Kappa particles to multiply, and also no synthesis of new Kappa particle production. So, this is what is the importance of sensitive strain is of. So, Kappa particles, as we have understood, along with the capital K dominant allele will bring about the specialty of Paramecium aurelia to behave as a killer strain. When either of which is not there, or when it is absent, it may lead to the strain called as the sensitive strain. Also, we find Paramecium generally showing a kind of reproduction called as parasexual reproduction or conjugation. So, conjugation is a process where two organisms of Paramecium aurelia are involved for a shorter time by forming a cone or a tube that junctions between two Paramecia. So, when two Paramecia physically gets connected, there occurs through this conjugation tube or cone the exchange of genotype, and sometimes when it prolongs you find cytoplasm also gets exchanged between the two Paramecia. So, conjugation is a false sexual reproduction that we find in Paramecium, or it is a primitive type of sexual reproduction that we see in the animals. So, here when a killer strain, when I say killer strain, you should remember this strain has got inside the cytoplasm killer particles or Kappa particles along with the genotype capital K in it, either in homozygous or heterozygous form. But here is a condition where the conjugation is involved with a killer strain. Condition is killer strain has got Kappa particles in the cytoplasm and the genotype is homozygous capital K. This is variety of killer strain is now conjugating with the other strain called sensitive strain, where the sensitive strain cytoplasm is devoid of the Kappa particles, at the same time they have the genotype of small k, homozygous condition. So, these two when involves in the conjugation process, there occurs the exchange of the nuclear material, and when they involve in the conjugation they are called as conjugants. So, after the process of exchange of their nuclear material they separate out. So, the separated individuals now are called ex-conjugants. So, importance of ex-conjugants is that one capital K, one small k will be present in each of these ex-conjugant. Why? It is because the nuclear genotype got interchanged or exchanged. One capital K from the homozygous capital K killer strain got moved into the sensitive strain with small k. Similarly, small k genotype, one of the allele has entered into the killer strain. So, killer and sensitive strains have exchanged their alleles of genotype that they have, only if it is for shorter period. Now, after the exchange of their nuclear material, the genotype of each of this ex-conjugant is one capital K, one small k. The ex-conjugant of the killer strain, when you look at it, forms the killer strain only. Why? Because it will have, even though the genotype is capital K, small k, it has, because of one capital K and the cytoplasmic containing the Kappa particles in it, it acts as killer. And genotype with capital K, small k of ex-conjugant sensitive strain that you see down towards right side, it forms sensitive strain only. Why? Even though capital K allele is there, the cytoplasm does not have the Kappa particles. So, what does it indicate? Only the nuclear inheritance, exchange got seen between the conjugates during the conjugation process. So, after the ex-conjugation, we will look into the nuclear content with the similar genotype, but the cytoplasm is different, thus they also differ with the type of strain that they are. I repeat, capital K, small k is common for both the strains of ex-conjugants, but one strain of killer will have in its cytoplasm the killer particles or Kappa particles. Cytoplasm of the sensitive does not have it. So, it remains a sensitive strain only. Now, these ex-conjugants further, when they undergo the binary fission process, they form the daughter cells or daughter organisms that differs with the character again. Now, with that little introduction, concept of conjugation we shall understand if it happens for a shorter duration which is lesser than 3 minutes of time. So, in Paramecium aurelia, conjugation takes place if for lesser than 3 minutes of time. Otherwise, in short duration when conjugation process is seen, there occurs only the nuclear material exchange as we have discussed earlier, and no exchange of cytoplasm absolutely is seen. So, this will indicate the nuclear inheritance pattern. So, what is happening here? The exchange of nuclear genes are seen between killer strain and the sensitive strain, and cytoplasm does not involve in the exchange. So, after the ex-conjugants are formed, they will have heterozygous capital K in them, that is killer gene in them, but the cytoplasm of the original parent is retained in them. So, thereby one cell will form the killer strain with heterozygous K and the other strain forms sensitive strain with heterozygous K. Now, the one of the ex-conjugant with killer strain of capital K small k, when it undergoes binary fission, it forms two daughter Paramecium as you see in the diagram down below of the slide here, and you find extreme left side capital K genotype is seen along with the Kappa particles in the cytoplasm. So, what is happening here? Of the heterozygous capital K small k, capital K is getting into one of the daughter cell and small k will enter into the other daughter cell. So, that small k with the cytoplasm having Kappa particles is forming sensitive strain. Why? Capital K allele is getting absent here in this strain. So, when you look at the ex-conjugant of killer strain, it will form one killer strain, one sensitive strain of the daughter cells. Whereas the sensitive ex-conjugant with one capital K small k when we look at, it will form a daughter cell with capital K genotype, but the cytoplasm is without the Kappa particles as you see down there, the third picture in the last row. So, indicates that the Kappa particles are absent in cytoplasm, so definitely it is of sensitive strain. The last one to extreme right is another strain with small k and cytoplasm having no Kappa particles, that also forms sensitive strain. Students, remember, ultimately how many daughter cells are formed? 4. Of the 4, 1 is said to be killer, rest 3 are said to be sensitive. The condition here is that only in the first individual capital K allele is also there along with their cytoplasm with the Kappa particles forming it as killer. Rest all does not follow the rule of it or conditions of it. So, they all remain as sensitive strain. So, when the process of conjugation occurs for a shorter duration, the nuclear exchange occurs and the number of killer strain and the sensitive strains when we look at it is 1:3. So, this is what the picture that depicts the conjugation process when it occurs for a shorter duration. The second condition we shall understand. Students, remember, if the conjugation occurs for a longer time, that is more than 3 minutes of time when it involves with, along with the nuclear exchange, the cytoplasm also gets exchanged. So, cytoplasmic content of the two strains will start exchanging between the organisms during the conjugation. So, that is what you see in the diagram down here. So, what is happening now? So, after mating, the sensitive strain also behaves as a killer strain. Why the killer strain has seen in the sensitive organism that is involved in the conjugation? It is due to the cytoplasm inflow in it and bringing in the cytoplasmic character in it. So, both becomes killer strains. Heterozygous, at the same time cytoplasm got exchanged. So, both are killer strains now. So, ex-conjugants here are both killer strains, but heterozygous condition. When both undergoes the process of the binary fission, we find capital K small k genotype with the condition of cytoplasm with the killer particles or Kappa particles will form a killer strain down, whereas small k with the cytoplasm having Kappa particles will form sensitive. The third organism when you look at here, that is also killer strain only, but with capital K genotype along with Kappa particles, when it gets into the daughter cell, it forms killer strain. And the last one, small k genotype, since it is present, even though cytoplasm has got Kappa particles, they will not form the killer strain. So, overall when you look at one killer, one sensitive strain of 1:1 ratio is getting formed from the from the parent ex-conjugant paramecium. So, this is the importance of the concept related to cytoplasmic inheritance of Kappa particles in paramecium arelia. So, this is the picture that depicts the conjugation process if it goes for a longer time. You find ultimately down the two ex-conjugants will be killer strains and two will form, each of it will form one killer, one sensitive strain as daughter cells down. So, that is about the importance of cytoplasmic inheritance of Kappa particles in paramecium. There is one similar such kind of process is seen in the higher group of organisms, where you have male and female difference. Students, in paramecium you do not have male and female difference, both organisms are involved there. So, we can have the option of male and female for any of the organism there, whereas, so there is specificity for the sexually reproducing organisms as males and females or father and mother is seen for the group of organisms of sexually reproducing group at the higher level. So, when there is specificity of a male and female, as we have discussed in the introduction, male will contribute to lesser cytoplasm and mother's ova will contribute to more or higher amount of cytoplasm. So, mother's genotype or phenotype will directly affect the phenotype of her progeny. In other words, the genotypic trait of mother is expressed directly in the child irrespective of the genotype whatever the child has. So, this type of effect is called maternal effect or maternal inheritance. Why it is? It is because of the genes present in the cytoplasm of the mother. Okay, so that is the gametes of the mother. A best example that we find here is relates to the shell coiling pattern that we see in water snail called Limnia peregrina. The diagram, the picture what you see down in this slide. So, the best example for explaining maternal effect is the direction of coiling or chirality of shells of water snail technically called Limnia peregrina. So, it was given first by Sturtevant in 1923. Students remember actually Boycott and Diver, where the scientists in the same year 1923 explained the coiling of shells types in the lemnia, but the pattern of inheritance was explained by Sturtevant. So, down in the picture you can see towards left side is the organism of lemnia, towards right side is the photograph of Sturtevant. According to the scientists, there are two types of shell patterns that are seen in the lemnia. One which coils towards the right side, one which coils towards the left side. The right side coiling of the shell is called dextral. Dextral in Greek refers to right, sinistral refers to left. So, the left coiling of the shell pattern we call it as the sinistral. So, dextral for right, sinistral for left coiling. This coiling of the shell, especially of the right side, is controlled by a dominant allele capital D. So, it is a nuclear gene that controls the dextral rotation of or coiling of the shell. So, the organisms with right hand coiling shows capital D or capital D small d as their genotype, and sinistrals will show small d as their genotype. That is recessive and dominant conditions overall. Now, your concentration should be on the cross. We are going to take a cross or involve a cross where the females show right hand coiling of the shell, males will show left hand coiling. In simple, they are following the pure line characters, homozygous dominant, homozygous recessive. So, mother is showing right hand coiling of the shell, father is showing left hand coiling of the shell. So, genotype is capital D for the mother and small d for the father. So, as you see in the picture down there, F1 individuals show heterozygous capital D small d genotype indicating dextral. So, this is a pattern of nuclear inheritance that we find according to Mendel. Now, when two such parents of F1 are involved in a cross, they form F2. In the F2, if you could concentrate, the genotype of capital D first towards the left side forms down, it forms dextral according to genotype. So, next organism is capital D small d, the second and third both are dextrals, very fine. Whereas the fourth individuals that you find towards extreme right in F2 generation is showing the genotype small d, but shows the character of dextral rotation, right hand rotation. So, father's character is no way involved in the child even though it has genotype small d in it. So, sinistral character it was supposed to show, but it is not showing. So, scientists later have found that the coiling of the shell is controlled not only by the genotype of capital D allele, but also the plasma genes that are present in the cytoplasm of the mother's egg cell. So, mother's characters are influenced here, whether it is F1 or P1, those characters are getting introduced into the child irrespective of the genotype. So, actually the last individual in F2 group down should be sinistral, but it is forming dextral. Why it is? Its mother is showing dextral character, so the dextral character is exhibited here. So, this is what you call it as maternal effect. What is maternal effect? The character of the mother is influenced or passed on or transferred into the child irrespective of the genotype whatever the child has. So, even the child has small d, it is not dextral or sinistral, instead it is mother's character that is dextral. right coiling it shows. So, this is what the cross that we have understood just now is of. Condition is when small d individuals are F2, when are inbred, when they are crossed with the other organisms, they start following sinistral individuals again, and capital D or capital D small d individuals will follow their nuclear pattern of inheritance. So, what is happening here? In the F2 generations, they are not able to express their character because there is a delayed effect of genotype that occurs in the maternal effect. Now, if we show the reciprocal cross, we are going to change the type of coiling between the mother and the father. Earlier we have taken mother's character as dextral, father's character as sinistral. Now we are going to have reciprocal cross. Mother showing sinistral, father showing dextral. In F1 generation we find capital D small d. What should be the character? According to Mendelian principle, dominant character should be expressed, it should be dextral, but it is not so, it is sinistral. So, we find maternal effect here. So, capital D small d genotype even though is present in F1, it is not showing the character of dextral, instead it is showing character of its mother, that is sinistral character. So, irrespective of the genotype, the mother's characters are entering into the child directly. So, this is called as maternal effect. So, similarly, when you see in F2 generation upon crossing F1, we find all the individuals are dextral, then in the later generation they retain back their own characters. So, this is what exactly is the concept of maternal effect is related. So, what is maternal effect? Condition where mother's phenotype or genotype will influence the character of her child irrespective of the genotype of the child. So, with this we are completing the concept of coiling of shell pattern in lemnia. So, earlier two slides again shows how the complete crosses, how it is between the mother when it is dextral and mother when it is sinistral in the next slide. Well, students, after we understanding the concept of maternal effect and cytoplasmic inheritance in paramecium, now let us answer the MCQs to know how well we have understood the concept. Question 1. Extra-nuclear genes or DNA molecules present in these cell organelles. Name the cell organelles in which these extra-nuclear genes are present. It is plasmids, it is mitochondria, it is chloroplast, and it is endosymbionts as Kappa particles, what we have seen. So, answer is B. Question 2. Some strains of paramecia contain Kappa particles in their cytoplasm with capital K genotype. So, dominant genotype along with Kappa particles if it is, what is the type of paramecium it is of? It is killer strain, it is answer A. The genotype of killer strains are capital K or capital K small k. So, answer is C. Dextral shells are rotatable towards the right side. So, which coil to the right, answer is D. When a cross is made between female snails coiled to the right, that is capital D, females or mothers are right hand coiled, fathers or males are left hand coiled. What is the result of F2 generation? F2 generation show all of them showing the coiling towards the right side. So, answer is A. So, outcome of today's session is we have learnt the cytoplasmic inheritance of Kappa particles in paramecium aurelia and maternal effect in the shell coiling pattern of water snail limnia peregrine. So, the references of today's session includes web references of Wikipedia and Britannica.com and book references of Textbook of Genetics by Ajay Paul, Genetics by P.K. Gupta, Introductory Zoology Volume 5 by C.M. Vimala and Principles of Genetics by P.D. Singh. Thank you all students.